Let's discuss what the evidence for evolution was. The discovery of genes as a mechanism to inherit characteristics and create variants supported the theory of evolution. There are also two other main sources of evidence, which were fossils. Fossils show the gradual changes in organisms over vast periods of time. And the other source of evidence is antibiotic-resistant bacteria. Natural selection neatly explains this phenomenon because bacteria reproduce rapidly, mutations can occur during reproduction, and if the mutated bacterium is resistant to antibiotics, it will survive exposure to the antibiotics and go on to reproduce. And this bacterium will represent the original ancestor of a new resistant strain. So they have evolved by natural selection. What are fossils? Fossils are the remains of traces of ancient life. They are found in rocks and have been preserved by natural processes. And there are three different ways of forming fossils. One is preservation when decay is not possible. Where decomposers cannot act, preservation is possible. So, for example, decomposers cannot operate in glaciers because this is an extreme environment with very low temperatures, so enzyme action doesn't really work. And also in peat bogs, which are very acidic, so the pH is too low. Another method of forming a fossil is the hardening of soft materials. The hardening of soft materials can produce casts or impressions, such as footprints. For example, clay hardens around an organism that is buried within it. This organism then decays, leaving behind a cast of itself. Another one is mineral replacement. Hard parts of organisms are gradually replaced by minerals as they decay slowly, creating a rock-like substance. When the surrounding sediment turns to rock, the teeth-shaped mineral structure remains distinct. So to recap, the three causes of fossilization that you need to know about are when decay is impossible, it can be preserved in peat bogs and glaciers. The soft materials can harden, and minerals can replace bone. So I'll have a little look at resistant bacteria now. So how do a strain of bacteria become resistant to an antibiotic? If a mutated bacterial pathogen is resistant to an antibiotic, when a person is treated with antibiotics, the bacteria will survive and go on to reproduce. This bacteria is the original ancestor of a new resistant strain. This bacteria will spread, as there will be no treatment for it and people are not immune to it, as they were previously treated with antibiotics. An example of an antibiotic-resistant bacteria is MRSA. The development of new antibiotics is a slow and costly process, and it struggles to keep up with the emergence of new strains of resistant bacteria. So we need to take measures to reduce the emergence of new antibiotic resistance. So what could we do? Doctors could avoid prescribing antibiotics unnecessarily, for example, for treating non-serious or viral infections. Patients must finish a course of antibiotics to make sure that all of the bacteria are killed. And the use of antibiotics in agriculture, for example in cattle feed, should also be restricted.